So here we have a concept that has come up several times within the first half of the course. And now we can think about it as it pertains to the cardiovascular system, and that is sickle cell disease. This is an example of an erythrocyte based disorder. As an overview from earlier discussions of this content, sickle cell disease is a result of a point mutation. So a single change within a nucleotide impacts the DNA, which impacts the mRNA, which impacts the codon in such a way that the amino acid that results will shift. The property of that amino acid goes from being polar to nonpolar. And as a result of that point mutation and amino acid change, the overall organization of the erythrocyte is affected. So when we think about this defect, it is a defect in hemoglobin protein. And recall that when it comes to an erythrocyte, it's essentially, because we've removed nucleus, we've removed organelles, is essentially just a sac of hemoglobin. So we have a defect in hemoglobin, and as a result of that defective hemoglobin, we are going to have a change in the shape of the erythrocyte so that it is a sickled shape rather than the canonical biconcave shape of a normal erythrocyte. And this is hereditary. So this came up again after the idea of point mutations, etc. This also came up from a genetic perspective. So it's hereditary. It is found in higher amounts in people of African and Mediterranean descent. When it comes to the mutated hemoglobin, we focus initially on its positioning within an aqueous environment. Now we'll consider the mutated hemoglobin behavior in oxygen deficient environments. So mutated hemoglobin doesn't bind to oxygen well. And so in locations where oxygen concentrations are low, hemoglobin becomes deoxygenated. Okay, so it's going to let that oxygen go. Now, what ends up happening with the hemoglobin is that it has a tendency to polymerize and it forms a gel. And that gel makes the erythrocytes elongated and pointy at the ends. Okay, so you can see the stretching out and then the pointy ends here. Now, sickled erythrocytes are sticky, and so they'll have a tendency to agglutinate, which you can see an example of right here. And so agglutination is essentially clumping together. Now, agglutination, when we think about the role that erythrocytes are meant to play and where they're supposed to travel, can be really detrimental to our bodies because agglutination can block small blood vessels, and that can lead to intense pain in individuals. It can lead to oxygen-starved tissues. And blocking circulation impacts effective blood flow. And if we don't have blood flow to the areas that we need, that can result in things like kidney failure, heart failure. It can result in strokes and joint pain and even paralysis. Now, when it comes to this process, there is hemolysis, which will destroy these cells themselves. And in an effort to do that, anemia results. And anemia is essentially a diminished hematocrit value, if we connect this back to our earlier discussion. Now, with that decreased concentration of erythrocytes and hypoxic conditions, and hypoxic conditions will mean we have low oxygen, that's actually going to stimulate sickling. So the process of entering areas where the oxygen concentrations are low induces the polymerization. Okay, so here we have decreased concentration of erythrocytes due to hemolysis because we're trying to get rid of these abnormal erythrocytes. That's going to result in hypoxic conditions because now we actually have less erythrocytes that are able to carry our dissolved gases. And that's going to stimulate the process even more. So unfortunately, in an attempt to fix the issue, we are making it worse. Now, if there is chronic hypoxic conditions, that can cause fatigue. So imagine we can't receive the oxygen to our tissues that need it. It can also impact mental development. It can result in deterioration of the heart and other organs. Now, in order to try to counteract the hypoxemia, there will be hematopoietic tissues that will activate, but the level of activation that is seen within those tissues actually causes bones to become enlarged and sometimes misshapen. So this can be a really harmful disorder. And it's also an example of pleiotropy, which is a concept from the genetics portion of the Campbell book. And that very briefly simply means that there are multiple phenotypic effects that arise from a change in a single gene. Now, if we think about all the things that are going on here, this seems really deleterious and doesn't seem beneficial at all. But in places where the parasite that causes malaria is found, these sickle-like shapes are useful because the sickle cell is detrimental to the parasite that would feed on it. And the advantage to this is conferred to the heterozygous individual. And the heterozygous individual is pretty resistant to the disease that would be caused by that parasite, which is malaria. So because of the heterozygous advantage conferred, the gene persists within a particular population. So when thinking about sickle cell disease, still think back to the earlier concepts. So the idea of changes within a polypeptide and how that change in a polypeptide impacts protein behavior. Now we layer that onto the idea of circulating through various blood vessels and the likelihood of agglutination, clumping together, and how that can impact effective blood flow. And then thinking about how ineffective blood flow means we have reduced capacity to exchange gases and how that can impact different areas of the body. When it comes to blood, we lose it continually. That can be through bleeding, through blood cells reaching the end of their lifespan, through using up components within the plasma, or through releasing components of blood through the process of excretion. And based on these events, one would expect there to be a need for continual replacement of these cell types in order for events that blood helps to support to be able to continue. The process of producing additional formed elements is the process of hematopoiesis. And the formed elements of blood trace their origins back to hematopoietic stem cells, often abbreviated as HSCs. And when it comes to HSCs, they are located in the bone marrow and are found in high amounts within the ribs, the vertebrae, the pelvis, and the sternum. And when it comes to HSCs, we will consider them to be multipotent, as these cells are destined to give rise to multiple mature cell types downstream. Now, a point related to describing HSCs as multipotent is that hematologists do not use this term in reference to HSCs. They will use the term pluripotent, but stem cell biologists utilize the term multipotent, and we are going to retain that classification for these cell types in our discussions through all topics related to hematopoietic stem cells within this semester. Now, in thinking about stem cells, one capability that they have is the ability for self-renewal. And we definitely want HSCs to have this property because we want to be able to give rise to all of those downstream types of formed elements. Now, when it comes to hematopoietic stem cells, there are two lineages that they can give rise to. So there's this myeloid stem cell lineage and this lymphoid stem cell lineage. And when we go one step beyond these lineages, we can see that there are progenitor cells. These progenitor cells are a little bit more specialized than that of the hematopoietic stem cell, and these are called colony-forming units. And each type of colony-forming unit, you can see that we have 3 here, are destined to produce some type of formed element downstream. Now, when we're thinking about the process of hematopoiesis, we can think about the formation of our formed elements. So red blood cells, megakaryocytes, which will give rise to these sheared components of platelets, all of our different leukocytes that we've discussed, and our lymphocytes, which are associated with that lymphoid stem cell lineage. But there's also the other components that we find within blood that needs to be replaced too. Blood's composed mainly of water, and it can obtain that water through absorption from the digestive tract. There's also acquisition of nutrients and electrolytes, and gamma globulins can come from connective tissue plasma, and proteins found within the plasma can be derived from the liver. So in thinking about this particular slide, some of the information has already been mentioned in the discussion of the immune system, as well as the stem cell content. But in thinking about this, we are just providing a general overview of hematopoietic stem cells and connecting the necessity of these stem cell types to the production of the components that we find within blood. Hemostasis is the natural process of stopping the flow of blood, and there are different hemostatic mechanisms. One is forming a platelet plug, another is blood coagulation or the formation of a fibrin clot. But there is one mechanism that's missing from this illustration, which is a vascular spasm, and that's the most immediate response to blood loss. So that would precede those other two mechanisms, and that's just going to be constriction of the broken vessel. So initially, the vessel gets broken, and then there will be a spasm within the blood vessels. And this is effective for smaller blood vessels, and it's useful because the spasm helps to reduce blood loss for like 20 to 30 minutes, and that helps allow for enough time for the other two mechanisms to occur because they take a little bit longer. Now, when it comes to this process, platelets are something that we discussed within the formed elements portion found within the Buffy coat, and they play a key role in hemostasis because they aggregate. So they kind of clump together and they'll form a plug that will temporarily seal a broken vessel wall. So that's the platelet plug here. Under normal conditions, platelets don't stick to each other, so they're not going to aggregate and they're not going to adhere to the endothelium of your blood vessel. And that's essentially due to chemicals that are released by the endothelial cells that inhibits platelet aggregation from occurring, because you wouldn't want to form clots or, kind of mesh nets, so to speak, in areas that don't need it. But when the endothelium is damaged, like in step 1, the collagen fibers become exposed and the platelets will begin to go through that process of adhering to one another. Now, as platelets aggregate, they release chemicals. Those chemicals help to induce aggregation of more platelets and so on and so forth. So this would be an example of a positive feedback mechanism that allows for the buildup of platelet aggregates that allows for the platelet plug to form. But when it comes to a platelet plug, this is not really sufficient at sealing something large, because when it comes to the platelet plug, while it isn't super obvious within the illustration, the platelets are not held very close together. So they're held pretty loosely together. So the final stage, being able to form that fibrin clot, is the result of a multi-step process that involves many factors. And so we can see here that there are clotting factors that are derived from platelets and damaged cells and plasma, and there's a ton of different factors that are found within a cascade. And when it comes to the process, it's simplified within this illustration. Within this simplified pathway, what we're focusing on is the formation of a prothrombin activator. And so that's essentially something that's going to come out of that enzymatic cascade, and it's going to act right here. And the role that prothrombin activator is going to play is that it's going to catalyze the conversion of prothrombin to the active enzyme thrombin. And when we have thrombin, we can see here that there's a positive feedback loop, helps with the cascade that leads to more activators. So that way we can amplify, build up the concentration of thrombin. And thrombin will further catalyze another portion within this process, which is the transition from fibrinogen into fibrin. And when fibrinogen transitions into fibrin, the molecules will form insoluble polymers. So this step helps to induce polymerization. And when we have that polymerization, the fibrin is going to act like a glue, and it's going to help to trap and keep all the platelets together. And that's going to prevent the exit of formed elements like the erythrocytes that are shown here. And when it comes to fibrin clots, these are really effective in sealing a hole or sealing a site of damage until a blood vessel can be permanently repaired. So in looking at this illustration, you have up here just a kind of showing red blood cells that are caught within threads of fibrin and the benefit of that in terms of impeding any excessive blood loss out of the blood vessel side. But when looking at this image, also keep in mind certain concepts that you've already learned about in the past, which is the role of essentially transduction pathways. Not quite in the same way as far as the way it's done within chapter 11, but just the idea of having a cascade of events that are dependent on the events that precede them, the concept of positive feedback. Here we have an example of changing something to an activated enzyme. And then when we go from fibrinogen to fibrin, this is different from that. We're not generating an active enzyme. We're generating fibrin, which is able to polymerize, and the role that polymerization plays in terms of helping to prevent loss or excessive loss of formed elements. So there's quite a few things here that do draw upon previous content, not so much from I guess not necessarily testing wise, right? Because I know that's sometimes the emphasis, but more in terms of helping to support your understanding of the events that are going on here. Here we go through cardiovascular disease, and cardiovascular disease are going to be diseases that impact the heart or impact blood vessels in some way. And as one would imagine, they can vary with respect to how serious they are. So there can be things that may be considered clinically to be minor, or they can be really essentially integral to life, right? So something that might impact blood flowing to really essential locations such as your heart or your brain. Obviously, we won't go through all of the different types of cardiovascular diseases. We'll go through a few, and for a few examples, we'll think about ways in which we can address or fix that disease or disorder. But even in ones where we don't expressly do that, think about some possible ways in which you could modify or change something within the cardiovascular system that might help to lower the likelihood of the event taking place, or some type of mechanism that can be used to treat that affliction. Here